Welcome, my dear students. Today we dive into one of the most fascinating and essential fundamental processes of molecular biology, that is DNA replication in prokaryotes. Just as a story continues, only when its text is copied accurately, life continues only when a cell can faithfully duplicate its genetic material. In prokaryotes like E. coli, this process unfolds through a beautifully coordinated series of steps involving a remarkable team of enzymes working with precision and speed. From the very first unwinding at the origin to the final sealing of the last nucleotide, every action ensures that the genetic blueprint is passed on without error. So let's explore this step-by-step mechanism and understand how the cell's molecular machinery safeguards life's continuity. Introduction. Prokaryotic DNA replication is a highly accurate, fast and well studied using Escherichia coli or E. coli. Replication is a semi-conservative and bidirectional process. It requires a large assembly of proteins acting together, often called the replisome. So this is the image showing bacterial chromosome replication. Here is the site named as origin of replication, from where the process replication is initiated. This is the replication fork. It is dividing, and this is the site where replication ends, and after the end of the replication process there will be two circular double-stranded DNA molecule. So from one circular double-stranded DNA molecule, two circular double-stranded DNA molecule will be formed, and this is known as bacterial chromosome replication. And this is the image showing the autoradiograph of an E. coli chromosome in the act of replication, the image showing the replication fork here. Now we should know about the origin of replication, which is the sequence named as oriC. Features of oriC: in bacteria or E. coli, oriC contains three key DNA elements. First one is the AT-rich region. This is the blue boxes are the AT-rich region. It is a 13 base pair long sequences. There are 13 base pair long sequences. AT pairs only two hydrogen bonds, so it is easy to unwind. So from the region within a DNA where the DNA replication will be initiated, it must be unwind firstly, because replication to initiate the process, the double stranded DNA must unwind. So AT pairs as have only two hydrogen bonds, it is easy to unwind. Second one is the DNA box. The yellow colored boxes are the DNA box. It is there are 5 DNA boxes within the oriC region, and each DNA box contains 9 base pair repeats, and it is served as the binding site for the DnaA protein. From the name of the DnaA protein, the sequence has been designated as DNA box. Third one is the GATC site. GATC sites are the underlined sites here. It is can be found within the AT-rich region or outside the AT-rich region. Also, there are GATC methylation sites. It is required for proper timing and regulation of the replication process, and the methylation process is occurred by the enzyme DnaB methylase. Now, this is the process, this is the step named as initiation of replication. First of all, role of DnaA protein. DnaA, this is the image showing the AT-rich region, and this is the DNA box. The DnaA proteins firstly bind to DNA boxes. This is the sequence where DNA box can bind. This is the yellow colored regions are the binding sites for the DNA box protein and to each other. Additional protein that cause the DNA to bend also bind. This causes the region to wrap around the DNA proteins and separates the AT-rich region. So here the DNA box protein binds and change the three-dimensional conformation and show the hydrogen bonds will be break in this region, in the AT region. So DnaA ATP binds specifically to DNA boxes, causes DNA bending and partial unwinding at the AT-rich region. Then proteins HU and IHF assist by stabilizing the bend. Then recruitment of the helicase. DNA helicase or DnaB protein binds to the origin, and then DnaC protein, which is not shown here, assist in this process. DnaC act as a recruiter and DnaB act as a DNA helicase. So DnaA recruits DnaC, which helps to load DnaB helicase onto the DNA. Two helicase molecules load, one for each replication fork. There is two replication fork, and for each replication fork there are two DNA helicase protein. DnaB or DNA helicase then moves 5 prime to 3 prime direction. This is 5 prime to 3 prime direction, and this is another 5 prime to 3 prime direction along the single stranded DNA and unwinds the double helix. Formation of two replication forks. Helicase unwinds DNA, creating two Y-shaped replication forks. This is the Y-shaped replication forks, and replication becomes bidirectional around the circular chromosome. Enzymes working at the replication fork. DNA helicase or DnaB, it breaks hydrogen bonds between strands. It uses ATP hydrolysis and produces positive supercoiling ahead of the fork. Then DNA gyrase or topoisomerase II, it removes positive supercoil by introducing negative supercoils. It prevents DNA from becoming overwound. Single strand binding proteins or SSBs, it bind to the separated strands of the DNA, and it binds the single stranded binding protein and prevent re-annealing or rebinding of the formation of the hydrogen bonds between the complementary strand, and protect the DNA as single stranded form and also from the nucleases. Then primase. Primase synthesizes the RNA primer, which is a 10 to 12 nucleotide long, ribonucleotide long, and it is required because DNA polymerase cannot start synthesis on its own. DNA polymerase in E. coli, there are three types. DNA Pol III, that is a major replication enzyme. DNA Pol I, that is that removes RNA primers and fill gaps, and DNA Pol 245, involved in DNA repair and translation synthesis. These are the different proteins involved in E. coli DNA replication, and their functions has been also given here. This is the function of key proteins involved with bacterial DNA replication. DNA helicase, we know, break the hydrogen bonds between the DNA strands. Topoisomerase II then alleviate positive supercoiling. Single stranded binding proteins or SSB keep the parent strands apart. Primase synthesizes an RNA primer, and DNA polymerase III synthesizes a daughter strand of DNA. Then DNA polymerase I excises the RNA primer and fills in with DNA not shown here. DNA ligase covalently links the Okazaki fragments together. This is the process, and the protein machinery of the DNA replication in cell has been given. Structure of DNA polymerase III. It is a multi-subunit enzyme, and it has different subunits like alpha subunit, which has the main polymerase activity. It synthesizes DNA. Beta clamp, it increases the processivity of the polymerase enzyme. Epsilon subunit, it has 3 prime to 5 prime proofreading activity. Theta subunit, it stabilizes the epsilon, and it is accessory protein that stimulates the proofreading function. Clamp loader complex, that is loads the beta clamp, and clamp loader complex includes the tau protein, gamma protein, delta, delta dash, phi and chi protein. So this is the image showing the how the DNA polymerase catalytic site, it binds at the catalytic site, and it starts the process of DNA replication. Directionality of DNA synthesis. Two key limitations of DNA polymerase: it cannot initiate DNA synthesis in vivo. So first of all, primer is needed to initiate the DNA synthesis, and it then can add nucleotides only in 5 prime to 3 prime direction. This image showing the direction of the DNA polymerase, that is in 5 prime to 3 prime. So DNA polymerase is unable to covalently link the two individuals nucleotide together. So primer is needed able to covalently link together. Leading and lagging strand synthesis. Leading strand synthesized continuously and requires only one RNA primer. This is the leading strand. synthesis. It is this is the leading strand synthesis and this is the leading strand synthesis. If only one RNA primer is there, DNA Pol III moves toward the replication fork. Lagging strand, it synthesized discontinuously. It requires multiple RNA primer, and DNA Pol III moves away from the fork. From Okazaki fragments, 1000 to 2000 nucleotides are found in bacteria and 100 to 200 is found in eukaryotes. DNA strands separate at origin, creating two replication forks. Primers are needed to initiate DNA synthesis. The synthesis of the leading strand occurs in the same direction as the movement of the replication fork. The first Okazaki fragment of the lagging strand is made in the opposite direction. The leading strand then elongates and a second Okazaki fragment is made. The leading strand continues to elongate, a third Okazaki fragment is made then, and the first and second are connected together. Now, we should know about the processivity of the DNA polymerase III enzyme. What is processivity? It is the ability of polymerase to remain attached to DNA during synthesis. And role of the beta clamp, it forms a sliding ring around DNA. It ensures Pol III does not fall off. It increases rate from 20 nucleotide per second to 750 nucleotide per second. Termination of replication, there are TAR sequences and TAS protein. It is located at opposite oriC region. T1 blocks fork moving left to right, and T2 blocks, this is the TAR sequence. Here the T1 binds and it moves the fork, it blocks the fork from left to right, and T2 blocks the fork from right to left. It ensures the forks meet at a controlled site. Final linking, DNA ligase seals final nicks and producing two separate circular DNA molecule. Catenin, for catenin formation and resolution, daughter DNA molecules become interlocked like a links in a chain. That is the after replication, there is a catenation formation. So how decatenation occurs? Topoisomerase action. It makes a temporary double strand break and passes one DNA strand through the break, and then resolves catenin. Two separate chromosomes are found. This is a separation of catenins. DNA replication can result in two intertwined chromosomes called catenins. These catenins are separated by the action of topoisomerase II. So, as we conclude or summarize, remember that prokaryotic DNA replication is a master class in coordination and accuracy. Starting from a single origin, the cell ensures bidirectional replication, flawless enzyme teamwork, continuous and discontinuous strand synthesis, rigorous proofreading, and final completion with remarkable efficiency. Understanding this mechanism is not just crucial for exams, it reveals how life maintains itself at the most fundamental level. With this clarity, you are now better equipped with your UG or PG and NET studies. Keep revising, stay curious, and continue exploring the wonder hidden within the molecular world. Thank you.